Good day, this is Professor Will, CBMD PhD, and today's Wednesday, March 9th, 2022. It is 2:12 and 18 seconds, a.m. Eastern Standard Time. Today's research, regenerating multiple cells. So let's hop to it here. The single protein prompts mature brain cells to regenerate multiple cell types. Findings can lead to new ways to treat brain injuries and disease. Date March 8, 2022. Source: UT Southwestern Medical Center. A single protein can reverse a developmental clock on adult brain cells called astrocytes, morphing them into stem-like cells that produce neurons and other cell types. The findings might someday lead to a way to regenerate brain tissue after disease or injury. Researchers are showing that it may be possible to reprogram the fate of this subset of brain cells, giving them the potential to rebuild the damaged brain. During development, mammalian stem cells readily proliferate to produce neurons throughout the brain and cells, called glia, that help support them. Glia help maintain optimal brain function by performing essential jobs like cleaning up waste and insulating nerve fibers. However, the mature brain largely loses the stem cell capacity. Only two small regenerative zones, or niches, remain in the adult brain, leaving it with extremely limited capacity to heal itself following injury or disease. Recent research has suggested that glia can be prompted to produce neurons in some models of brain injury or after genetic manipulation. Although these findings are promising, regenerating healthy brain tissue will require production of multiple cell types rather than only neurons. Looking for ways to spur this multipotent regeneration, the laboratory used a genetic engineering technique in adult mouse brains to induce astrocytes, a subset of glia, to produce different transcription factors, proteins, pivotal for controlling cell identity. And these experiments showed that a single transcription factor, a protein known as DLX2, appeared to reprogram astrocytes into neural stem-like cells capable of producing neurons and multiple subtypes of glial cells. The researchers confirmed these findings, both using a technique called lineage tracing, in which they followed progeny of the altered astrocytes as they multiplied, as well as marker analysis that showed that these cells had the expected identities of neurons or glia. Researchers showed that prompting astrocytes to produce DLX2 appeared to reprogram them into stem-like cells, with features of both immature brain cells found earlier in development and cells found in the regenerative niches of the adult brain. Researchers suggest that DLX2 might someday be used as a tool to treat traumatic brain injuries, strokes, and degenerative conditions such as Huntington's disease. Researchers are planning to study this approach in animal models. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.